Hello students, this is experiment number 14 to comment on the exercises of hybridization, emasculation, bagging and tagging. First, write the objective or aim of the experiment, that is to comment on the exercises of hybridization, emasculation, bagging and tagging. Then write requirements. We need setups showing emasculation, bagging and tagging, and for arranging these three setups we need ornamental plant, scissors, clip, brush, needle, plastic bag and forceps. Next, write the theory. Actually, plant breeding is a science where we change and improve the genetic makeup of plants and produce new varieties. And for plant or crop improvement, one of the major methods used by plant breeders is artificial hybridization. What is artificial hybridization? It is a method by which improved varieties of economically important plants or crops are produced. Here, crossing is done between two genetically different plants. Now, the main steps involved in artificial hybridization. They are: number 1, selection. First of all, select the healthy plant with desired character. And then, second step is emasculation. In this, anthers are removed by using forceps or scissors. This step is done before the maturation of stamen. With this, your first setup is ready, having a flower from which stamens are already removed. Third step is bagging. This emasculated flower is now covered with a bag of suitable size to prevent contamination of its stigma with unwanted pollen. कोई भी अनडिज़ायरेबल और अनवांटेड पॉलन ग्रेन्स स्टिग्मा पर न आ जाए, इसलिए इमेस्कुलेशन के बाद फ्लावर को कवर कर दिया जाता है बाय यूजिंग अ बैग. इमेस्कुलेटेड फ्लावर कवर्ड विद अ बैग इज योर सेकेंड सेटअप. फोर्थ स्टेप इज पॉलिनेशन. जब स्टिग्मा रिसेप्टिव हो जाता है, देन द पॉलन ग्रेन्स आर टेकन फ्रॉम डिजायर्ड प्लांट and dusted on stigma, and this is called as pollination. After pollination, flower is rebagged and fruit is allowed to develop. Fifth step is tagging. After controlled pollination, a tag is fixed below the flower which contains certain information like name of parent plant, date of emasculation, date of pollination, and also you can write name of plant breeder. A flower covered with a bag having information written on tag is your third setup. Note one important point. If female parent is unisexual, then emasculation is not required. This step is required in case of bisexual flowers. Now, all three setups are ready and you have to identify and comment on these. Setup A, as shown in figure. It shows the emasculation step. It is a step of hybridization. It is a process of removal of anthers and it is done to prevent self-pollination. It is done with the help of scissors or forceps, and also unisexual flower does not require emasculation step. So, write these points in comments of setup A showing emasculation. This is setup B. It is showing the bagging step. It is a step of hybridization method. It is done after emasculation to avoid cross pollination by unwanted pollen grains, and it is done on female flower, and after pollination the flower is rebagged. Setup C, it is showing the tagging step of hybridization. Here we have to write the details of experiment on our tag like name of selected parent, date of emasculation and pollination, and also you can write the name of plant breeder. After writing all these comments of 3 setup, draw neat and labeled diagram of all steps in your file. So that's all for today. Thank you very much for staying with me. Subscribe the channel to get the information about my new video. Thank you so much.